Welcome to the SPNHC ICOM NATHIST 2020 virtual conference. This is general session for Miscellany. I am your moderator, Susan Butts, and my helper is Kimberly Cook. This session will be recorded for later viewing. Thank you all for joining us, and thank you to all the speakers in this session. Each presenter will present for 10 minutes. There will be 5 minutes of questions at the end of each presentation and the time to transition between presenters. Please ask questions of the speakers using the Q&A feature found at the bottom of your screen, and these will be asked of the presenters by the moderator in the order that they were received. The chat function has been made available for technical questions or for conversing with other attendees. Please use this judiciously, as any nefarious or inappropriate use of the chat may result in you being removed from the session or the chat function being disabled. Please see our code of conduct document for more information. And please bear with any technical difficulties we may have and enjoy the session. I will be sharing my presentation first. And this is the final report for the Yale Peabody Museum's contribution to the Cretaceous World Thematic Collections Network, which aim to digitize fossils from the Western Interior Seaway. Waiting. For some reason, it's not going to the next slide, so let's just try this. All right, the Western Interior Seaway was a shallow sea that split the North American continent in two during the late Cretaceous, which is about from 100 million to 66 million years ago. And this time period is characterized by some of the highest temperatures and greenhouse conditions and high sea levels that we've ever had in geologic history. And scientists predict that we will experience global temperatures similar to the late Cretaceous in the year 2051. This makes the Seaway a particularly interesting laboratory for scientists seeking to understand the relationships between climate, biodiversity, and ecosystem stability over time. The Western Interior Seaway supported diverse ecosystems of microorganisms, invertebrates, and vertebrates, resulting in an abundant fossil record that has been heavily collected for over 100 years. While the fossils of the Seaway are well known taxonomically and very well represented in museum collections, their associated geographic and temporal locality data remain largely locked away on paper specimen labels in museum drawers. The Cretaceous World Thematic Collections Network was funded by the ADBC program at the National Science Foundation, and it has 9 partnering institutions listed here, with University of Kansas Biodiversity Institute being the lead organization, and with 2 unfunded federal partners, the Smithsonian and the USGS. And the overall goals for the project were to digitize IP, VP, and microfossils from all of the partnering institutions. We estimated that this would be about 164,000 specimens from thousands of localities that would be digitized, including georeferencing and imaging. And the minimum goals were exemplar imaging of 1,600 taxa, often from multiple orientations. And we wanted to share that data for research and education on iDig Bio and other portals. This TCN will generate new data that will enable scientists to answer novel questions about the interaction between diversity of species, ecosystems, and environmental changes during this critical interval of Earth's history. Beyond research, we wanted to introduce science students to the role of museums in scientific research and bring collections to the public, particularly K-12 students, through an educational portal and internships. Yale has a well-known history of research in the Western Interior Seaway and has many iconic specimens from the Western Interior Seaway, including Archelon, shown here with the collector, the fossils of O.C. Marsh, who's in the middle of this back row, shown here with the student members of his 1870 fossil expedition, the 16-foot-long Zifactin, a specimen collected by Sternberg. And the IP collections are very well developed by Carl Wagge, who is an IP curator and former director who collected fossils from the Seaway from the 50s through the 80s, along with many of his graduate students, including Neil Landman and Ian Speeden. And IP has one of the most comprehensive libraries of Western Interior Seaway publications and over 400 late Cretaceous ammonoid types. Specifically at Yale, in vertebrate paleontology, we had 1,663 catalog specimens that needed georeferencing and imaging, and approximately 300 uncatalogued specimens. In invertebrate paleontology, we needed to increase the robustness of about 7,000 cataloged objects we have, plus we had 134 drawers of material in the stratigraphic collection, which were uncatalogued but identified with locality information with the specimens. So this presented some problems. There's a huge disparity in specimen sizes, literally from microfossils to mosasaurs. And a good deal of the material is locked in these mineral concretions. So you can see these mineralized blocks that are just full of all different sorts of fossils. Clearly, we needed to be creative in developing some high throughput protocols. And what we decided to use was InSelect, and this was developed by the Natural History Museum, and it's freely available to catalog whole drawers of butterflies, and it automatically selects the butterflies and saves them as crops. That wouldn't work for our concretions, but you are able to manually draw boxes on the specimens. And here you can see this concretion, which is a high resolution image done with stacking photography. And a student has gone in and drawn boxes around each of the specimens in there. Then those are saved as crops, which are numbered. And you can see on the right, you can actually enter metadata. So if you wanted to catalog specimens at this stage, you could do that and add it into the metadata, and it would be incorporated into the CSV file that's spit out from InSelect. So InSelect extracts the boxes and they become individual numbered crops, and it generates this CSV file. We manually catalog the concretion itself, then run a series of scripts that apply a new string of catalog numbers to all the objects and other objects in the concretion, batch rename the numbered crops with the specimen numbers, and annotate images and upload the images to EMU, our collections database. And we have an extra QC phase, which is Clam Day, in which Jessica Utrup, a museum assistant at the Peabody, goes through and confirms the identification of all of the individual crops. We're also doing this for microfossils, so we're imaging in the lab of Pincelli Hull at Yale. And we have a couple different formats of slides, either numbered boxes or a strewn slide where the specimens are just tapped out onto a reversible adhesive. And you can see, as Chelly Hull puts it, endless forams, most beautiful. What we do in this case is we save a contact sheet, and we can find specimens again using the coordinates, which are also saved in InSelect. We've also tried using InSelect for vertebrate lots. So we'll divide up the lot and spread it out onto the table, and then use InSelect to catalog the material because it's all from the same locality and same identification. It's actually much faster than digitizing each item individually. And you can see these rapid throughput protocols have been really successful in getting a lot of material recorded for us, digitized for us. We've done 110,000 specimens, 110,000 lots, which corresponds to 160,000 specimens, which, if you remember, is consistent with the goals for the entire project. So that's great. We've done almost 98,000 images, many with multiple orientations, and we've georefed 2,600 collecting localities. And we've been able to have this support base, or this success based on the number of students we have working, our protocols, and we have students from Yale and beyond. We also have Evolutions interns, so this is the high school program at Yale at the Peabody Museum, and we've had students helping digitize, helping with 3D imaging, and helping with students in the Peabody summer camp. We've also created Cretaceous World, and this is cretaceousworld.org. And you can see that this is a K-12 portal for collections studies by students. And we've worked heavily, this is developed by Whirligig, and we've worked heavily with teachers and with the high school EVO program to make sure it has really good usability and is something that teachers actually want in their classrooms. So when you go there, it automatically puts you in browse mode rather than making you do a search with terms. You can filter and doing faceted searches rather than entering specific terms. We've also adapted it so that you can search by common name, and these are harvested from EOL. And just like you would get in any other collections database, you've got images, you've got all the relevant data that goes with it, you've got a small map that shows you where the specimen is from. And you can also, there's also tools for annotating and measuring. And the beauty of it is that when you register for Cretaceous World, you can link students and teachers together so that you know you're able to see what students are doing, you're able to send collections to them or have them send collections to you with their annotations and measurements. And so it's a great way to interact with students about biodiversity data. We also have material on Morphosource under the project name Western Interior Seaway. So I encourage you to look at that. Our material is also on iDigBio, so I encourage you to use that data, those data. And I'd like to give a shout out to John Hendricks and Bruce Lieberman, who created this Cretaceous Atlas of Ancient Life, featuring all of the fossils of the Western Interior Seaway. And most of all, I'd like to give thanks to the National Science Foundation, who funded this, and to iDigBio for hosting all the data and for being so proactive in getting the community together and on the same page and enthusiastic about this. Thanks. And I think we can ask some questions now.
There is one question in the Q&A. Kessler asks, how do your TCN partner institutions distinguish TCN-specific specimen records in iDigBio?
That's interesting, and that's something we've gone back and forth on, and I'm not exactly sure what the end result is. I know that there are certain fields that get populated, and I think we've just designated, so at the local level, at the Peabody, we have this seldom used field in EMU where it gets populated, everything gets marked as the collection National Science Foundation Cretaceous World TCN. So it's marked as a collection there, and then we have collaborated, all of us have collaborated with iDigBio to make sure that that is recorded, but I'm not sure exactly what the final result was.
Cool.
Yeah.
Allison asks, could you please provide more explanation on your lot imaging method?
So what we're actually doing is laying out these, so you have a tray of specimens, and we just spread them out. There can be some overlap of where the boxes are, that's fine. But we just lay them out, and generally with the specimen label and with the scale bar and a color bar if necessary, and simply draw boxes around them in the same way you would with within the concretion or with the microfossil slides. And then of course it saves those as crops, and we copy the data for all of the crops as the one specimen that's been already cataloged in that lot.
Awesome. Looks like there are no more questions. We still have, oh, there's one. McKenna asks, are all of the specimens you're digitizing from historical collections, or is there still active collecting going on in the region of the former Western Interior Seaway?
We have not received specimens recently from the Western Interior Seaway. I assume that there is some collection, probably Neil Landman from the American Museum of Natural History is one of the most active people. But we haven't, most of the stuff we have is from the Carl Wogge era, so from the 50s to the 80s. And we have like literally tons of material from that time period, from that time span.
All right, looks like, oh, just as I think that they're all over. Nisha asks, how long did it take to digitize specimens that you have so far?
Oh, so we're in a one-year no-cost extension. We worked for 3 years. And honestly, most of the digitization was done within 3 years, but there's always a lag time in the quality control, like making sure that the identifications are right, uploading all the pictures. It's like super time consuming to do that. And we'll usually save it as batches, but you know, those batches get bigger and bigger, and then we have to you know, spend days uploading material. But about, well, let's just say 3.5 years to do all of those. And that's working with, maybe 3 or 4 students during the school year working 8 hours a week, and 3 students in the summer, average of 3 students in the summer working 20 hours a week, plus a full-time museum assistant devoted to the project.
That's awesome.
Yeah.
All right, well, I'll answer this one last little question, then we'll switch over. From Richard, what type of tape do you use for your microfossils, and how would you remove individual specimens if needed?
We've used several forms. So the curator favorite is actually glue stick, which is apparently archival and reversible. And then we've experimented with several other glues, and you should send me an email and I'll tell you the ins and outs of all of the different forms. But I think we've used B72, which we didn't prefer. And then there's a proprietary name, and I can't remember the name of it. If Jess or Alyssa know, you could stick it in the chat. But send me an email, and I'll give you the full details.
Awesome.
But the idea is that we do want to make it reversible. A lot of these specimens are analyzed for geochemistry. And so if you find a particular specimen, we have to be able to go back and find that thing on a microslide and be able to get it off for you to grind up and use for geochemical analyses.
Susan, do you guys ever use gum tragacanth? Because that's what
we don't anymore.
For our microfossils on this project at sea.
Yeah, yep. And Jessica, it wasn't the UV glue, that was for a different project. So I'm not sure, you'll have to send me an email. It was a curatorial grade glue that was recommended by our conservators.
Cool, I think that's all the questions we have. And we'll go ahead and switch over to Robert Minckley and then start again at 10:50.
Okay. I'll give you a couple minutes, Robert, in case people are coming right for the, I'm at 10:48 right now, so.
Oh, okay.
I'll introduce you at least. So our next talk is Robert Minckley. His talk is The Ward Project in Ward's Natural History Establishment, Rediscovering the Museum Builder of America.
Should I go ahead and go?
Give it 1 minute.
Okay.
I'll say go ahead.
And I'll just remind people, if you have questions for any of our speakers, you can enter those in the Q&A at any time, even after the speaker has gone. The speakers will still be able to respond to your question in the Q&A. So if you think of something later in the session for one of our previous speakers, just pop that in the Q&A.
All right, Robert, why don't you start and I'll give you a 3-minute warning.
Okay.
Time. Great, thank you.
Okay, my name's Bob Minckley. I'm at the University of Rochester. And I want to talk to you about a project we've fallen into. In the 1870s, the 3 largest natural history museums in North America were at Harvard, which is Boston there on the right side of that map, at the Smithsonian, which is not on the map, and then in Rochester, New York, at the University of Rochester, which that large arrow was pointing to. Next slide. The sole reason, could you give me the next slide?
Yeah, it's just taking a second.
Okay.
Yeah.
The sole reason for the University of Rochester Natural History Museum was because of Henry A. Ward, who will be popping up here, a Rochester native born in 1834. At the age of 19, Louis Agassiz visited Rochester, New York, and met Ward, invited him to work with him at the Museum of Comparative Zoology. Ward accepted, but shortly after getting to Boston, quit so he could go to Europe and the Middle East, where he enrolled in the Paris School of Mines. He began collecting and selling fossils to support himself. And then importantly, he attended the Paris Exposition in 1855 And the Paris exposition was wildly popular. It attracted over 5 million people to the various exhibits. Next slide. Ward returned to Rochester in 1859, joined the university as a professor, and shortly thereafter established Ward's natural science establishment. At the age of 27, he opened his workshop of primarily fossils, casts of fossils and minerals to the public to considerable fanfare. He lasted only 7 years on the faculty and for the rest of his life devoted himself full-time to his business. The timing of Ward's opening was fortuitous. It was just after Darwin published and the world was absorbing The Origin of Species, and after the US Civil War, when natural history education was becoming incorporated into school curricula, and an explosion of natural history museums were being established, particularly in the US. In many ways, this period was the beginning of formal teaching of the natural sciences, broad-scale awareness of biodiversity, and recognition that humans had a role in biodiversity decline. Next slide. Ward's natural science establishment grew to be a massive company that sold anything from individual specimens to private collectors, to cabinets, to entire museums. They sold zoological, botanical, mineral, and fossil specimens, as well as models of things like early vertebrate development. To give you an idea of how big the company was, to prepare for the 1893 Columbian Exposition in Chicago, Ward's had 74 employees preparing specimens, and they filled 10 complete train cars of material. After the exposition, the entire exhibit was bought by Marshall Field, and that was the foundation of the Field Museum. Next slide. However, the first large sale by Ward's was to the University of Rochester in 1862. The collection was used by the university for public education, research, and classroom instruction. The geological material was arranged chronologically, and extant species arranged by evolutionary relationships as they were understood at the time. Next slide. The University of Rochester Natural History Museum is no longer active. However, much of the geological collection and parts of the zoological collection are still owned by the university. 6 years ago, the biology department curated the zoological material and databased it. What was left of it and the associated models. During that process, we contacted the archivist at U of R, which is Melissa Mead, and remarkably found out that not only were all the correspondence, catalogs, and bulletins associated with Ward's company from its inception, but also were in the U of R library, but also the diaries of Henry A. Ward, some of the employees and many members of the Ward family. In total, there's 24 linear feet of documents. Next slide. So the Ward project was born, and it represents an unusual but fantastically productive collaboration among the library, biologists and geologists, and staff in Digital Scholarship Lab, especially Josh Rumpf. As soon as the collaboration began, we were intent on establishing a portal that would collect any and all specimens and correspondence associated with early Ward's business. Initially, we thought we would do this by contacting museums and expecting them to respond to us. That plan was obviously overambitious and probably fatally flawed. Can you skip to 2 slides ahead right now? Later, we realized a better plan was to build a virtual pre-1920s Ward natural science establishment that would include digitizations of the bulletins, catalogs, and specimens that the company traded in, as well as correspondence and diaries. Next slide. The bulletins and catalogs are widely available and show how Ward's advertised itself. Some catalogs were used as textbooks at the time, so could have been considered outreach in today's terms. Unique to the copies at U of R, as you can see on the left there, is because they were used by the company, many of them are heavily annotated. Next slide. The specimens provide insight into the breadth of the material sold by wards and their attention to presentation. Just look at that armadillo there. Taxidermy specimens, skeletal materials, fossils and minerals, casts and models were all sold. Next slide. For many years, Ward's was the only company that Blashka glassware was sold through. Next slide. Oh, actually go 2 slides now. Why don't you go? Oh, I'm sorry. So the correspondence and diaries reveal the inner workings of the company. Conversations among scientists, among collectors for hire to supply the specimens, collectors building private collections, academics acquiring teaching material, and others that were building entire museums are all housed there. Go ahead to the next slide. Whoops, we're ahead. Can you go back? Let me go back to one more, sorry.
Got about 1 minute.
Okay, our goal with the website is not limited to making available the Ward's holdings at the University of Rochester. Rather, we designed wardproject.org to be a virtual museum that links our holdings to the materials held at other museum institutions. Ward material is in museums worldwide. In the late 1880s, they sold to approximately 250 institutions. 69 of those remain active. So next slide. So to finish, the goals of Ward's were to build museums that enhance natural history education and biodiversity awareness. Our portal will serve to recapitulate those goals. We've set up the virtual museum to build a site that can be visited by anyone, anywhere. We have the capability for others to contribute by transcribing letters or adding their own Ward material. Next slide. Specimens can be examined in a 3D viewer from any angle. We'll be adding soon a feature where you can annotate the 3D images. Next slide. Finally, the website uses Dublin Core and Darwin Core standards throughout. And really what links all these layers of the website are the scientific names, either of the specimens, the names used in the catalogs, the names in the letters, and that's all linked together using Specify. So the website is our museum's focused on one company, but one that was pivotal in the early growth of natural history education and expansion of natural history museums. So by linking the history of the specimens, collectors, and adventurers, it would be a valuable resource for the natural history museum community, as well as enthusiasts, historians, teachers, and other scholars. Thank you very much.
Thank you, Robert. That was great.
All right, we've got a couple questions in the Q&A. First, we have from Siobhan. What platform did you use to transcribe the letters and documents? Were they transcribed by volunteers?
Well, yes, you can sign up, and that hopefully someday will be a, it doesn't matter. Yes, it's set up so you can you can actually do it, you can volunteer and do it, yes. And the website is all Omeka based. Oops, there's the last slide, sorry.
All right, so Cyrus says, here at Harvard Museum of Comparative Zoology, we have many specimens purchased from wards in both invertebrate paleontology and vertebrate paleontology. Mm-hmm. Many of these have only vague locality info, such as Europe. Is there a possibility there's paperwork on purchases made by Harvard that may give more info or for georeferencing these purchases?
Absolutely. Well, in the letters, there's a tremendous amount of information. And we've got about 1/3 of the correspondence scanned now. It's not all publicly available on the website yet. And we're hoping to get the other 2/3 done in the next 2 years. So there is information there. The locality data is the problem with the specimens still that are here at U of R, and with the specimens that they sold. If you look in the bulletins early on, you can tell they were sticklers for recording locality data, but at least the specimens here, all that locality data is gone, and it's probably because they moved the specimens several times from when the museum was founded to where they are now. Maybe we can recover some of that from the letters. That would be great.
Great. Greg is saying that they suspect nearly all of us have ward material. What sort of information would you like from us, the community, and how should we get it to you?
We have all of the outgoing letters, but you have the letters that came from you back. So we're very interested in that. We want to have it so that images of your specimens with the ward labels, and even sometimes if you just have ward labels without the specimens, if that could be put on, or at least linked to our website, we've set it up. We've been working on this for about 5 years, so we've got it set up to be able to accept that. We don't have the person power to all of you send us your data this week yet, but we certainly, that is the goal of the website, is to be able to collect that and have it I don't know, coalesced in some sense, and so people can learn about what wards did. So contact us, please.
Great. All right. Laura is asking, at Michigan State University Museum, we have multiple articulated skeletons and other specimens with original Wards Institute labels affixes and some kind of numbers. What would be a good way to look up information for these numbers?
Yes, those numbers. So I'm going through all the catalogs now and putting the names and specify, and it's not obvious to me that those numbers were maintained all the way through the life of the company. I mean, the company's still here, but we're basically sort of interested in up to 1920 or so. And I don't have a good sense of whether the numbers in the catalogs, which probably are the same numbers that you have on your labels, were maintained all the way through. And so that's just something that I'm going to have to figure out. But I'd be very interested in see those numbers if we have to just start cross-checking them.
Great. Haley asks, did Ward receive trades from others, or did the company only sell specimens? They've looked at your project there at Amherst College and they have lots of data to add.
They did trade. They didn't just sell. I don't have a good sense of how much trading there was, but you can often tell that they were, well, I wouldn't say I've seen letters where they were trading from like the MCC and we'll give you these specimens if you could give some of those back, something like that. So I know that there was some barter going on, but it was primarily sort of bought and sold. So yes, we know about the Amherst collection. We have letters, we have some of those letters, and I think you've been in touch with us.
Great. Let's see. Have you noticed any telltale signs of Ward specimens, either techniques or materials that are unique to Ward specimens?
Yes. Well, I mean, Carl Akeley was a Ward employee, so I've become very interested in the preparation development of specimens. And some of it was here, some of it was elsewhere, but And yeah, I'm getting so I can recognize the mounts sometimes, that it's a ward specimen if the label's gone. And we're becoming pretty interested in sort of developing that as part of the website. Is you know, this is what they did distinctively. If you read in the bulletins, they talk a lot about, and the bulletins are all up now, they talk about specimen preparation and techniques. Yeah.
Oh, how much does Omeka cost? It's a big question.
Free.
Aha. Great. Let's take one more question. Let's see. Have you adopted new exhibition strategies based on new museology in your museum?
I'm not sure what that, you'll have to explain more.
Okay. Wait for them to put that in the chat. All it's like we're time for questions. This is sort of our built-in time for transitions, unless you have extra time. But
contact me. I'm glad to talk more.
Yep. Great.
That was great. We have so much wards material at the Peabody Museum, so I can't wait to talk to you more.
Yes, that's a good point. Robert, you're more than welcome to answer the Q&As that are still in the Q&A right now as we're switching over. So thanks. All right, next we have Wouter.
Okay, we'll give it 1 or 2 more minutes. 1 minute.
Thanks, Susan.
Mm-hmm.
Can I start?
Let's wait 1 minute and then I'll give you an introduction.
And if you could speak a little bit louder, a couple people mentioned in the chat in the first session that your volume was very low.
Okay, I will try. I have my volume not max, so.
Okay, thank you.
Okay. All right, so for our next talk in the session, we have Wouter Addink, and his talk is ELViS, the European Loans and Visit Systems. Thank you.
Thank you. This talk is entirely different from the previous 2 talks. I hope that you all find it equally interesting. So this is about the European Loans and Visit System. Next slide, please. So the European Loans and Visits System is a system that is currently under development in the Census project, which is funded by the European Commission. And it is going to be a future service in DISCO, the new research infrastructure for scientific collections in Europe. It will be an integrated system for giving access to the collections in Europe, and will support loans, visits, and applications for digitization on demand, and will track the outputs of these applications. So it is an open system that will provide free access for everybody in the world to the collections in Europe in a unified way. We started development end of last year. The first product that we developed was a minimum viable product, so it is a little typo here. That was launched in February, that just had the minimum functionality that was needed to launch the first virtual access call in the Census project. And virtual access in Census means really digitization on demand. So instead of getting to the museum to visit the collections or asking for loans, you can use this call to request digitization of collection items that have not yet been digitized on demand. And the results of these digitization requests will be published online. So the first call opened in February, and it's just closed. And we are now working further towards developing the first version of ELViS, which will be ready early next year, and should then be able to support not only the next virtual access call, but also a transnational access call. And you can use that to apply for a visit from another country to collections. We also will then have an help desk in place. Then we intend to leave the software a little well for usage and to see what improvements need to be made further. And then we will start a second round of development for towards ELViS version 2. And that version will be ready early 2022 and will support individual loads and visits. It will also have pilots for authorization access, and it will include the integration of the digitalization dashboard, one of the other services that is being created in the Census project. Next slide, please. So I would like now you to show how the virtual access requests works. I cannot do a live demo, so I will just do screenshots. But the benefit of that is that I can show you the full workflow for the virtual access. The first step is that you need to register as a new user. That's a pretty standard procedure. However, you will see here that one of the things that we require if you register as a new user is to supply your ORCID identifier. So you need to have an ORCID identifier in order to register and create new requests. We are not using the ORCID information yet, but we want to use that in the future, as this is a unique way that you can provide information about yourself, about the literature that you created, and about the research that you are doing. You are in full control of your ORCID identity, so to speak, and we want to use that information to provide information for the institutes to know how they need to prioritize your requests. Next slide, please. The second step is that you log in, and then you see that there is an open call for VA requests, and you can just click the button to create a new request. That will open a form that you then can start to fill in. Next slide, please. So this is how it looks. There are a few steps of the form that you need to fill in. You start with giving your request a title and a short description, and then you select the institutes and the collections at the institutes that you would like to include in your request for digitization on demand. There is a drop-down list of the institutions that are offering access to virtual access, so you can just select these. And on the right side, you see a comments section, and this you can use for entering additional comments. And later in the process, this can be used by others as well to have kind of a chat functionality about your request while it is progressing. Okay, next slide, please. So once you have been working on the draft request and you have filled everything in and you think it's ready to be made public, then you can submit it for evaluation. This will mean that the submission becomes publicly available for the virtual access coordinators of the institutes that you institutions that you selected, and they can then look at your request and start to comment on that. Maybe they want to have some additional clarifications, or maybe they say, oh, some parts of your request are not feasible and you need to change this a little bit, etc. And for that, they can use the comments section. Other requesters in the system will also see your request, and that's one of the beauties of this system, is that it really allows for community collaboration on creating these requests. So other requesters can see your request and can see if that matches with maybe with their request and whether these needs to be merged. Other institutes can maybe suggest also additional collections that you didn't think about to include in your requests. Okay, next slide, please. So in this step, once you have submitted for evaluation, the VA coordinators will be notified by email that there is a new request, and then they need to evaluate that. If your request involves several institutes instead of one, there will be several VA coordinators that will be notified. Initially, the stages will then be evaluated, and once one or two of the VA coordinators have approved your request, the status changes to being handled. Once all the VA coordinators of all the institutions involved have approved your request, then the status becomes approved. Next slide, please.
Got about 3 minutes.
Okay. So there are 10 steps in total, I think, and this is step 6. Yeah, as I told you, the VA coordinators can comment on your requests. Like you here see, there are some comments. What's the proposed timeline for data release? And you can answer to that to the same comments section. Next slide, please. And if you have made the required changes, then you can update the requests. Again, with an update button. Next slide, please. And then what the VA coordinator also needs to do is to add a cost calculation for the institution. So there is an upload part in the form for that, and the VA coordinator approves your requests. Next slide, please. So once all the VA coordinators have approved, the status becomes approved and the comment section will be closed. So your application is then final, your request is then final, and there is no longer an option to comment on that. And then the last
Wouter, about 1 minute.
Yep. The last step is that there is an admin that can download the requests and the cost calculations as a zip file for a prioritization panel that will then prioritize the requests based on the budget that's available in the project. So that's the last step. And that's the full virtual access process. So that's the most important thing that I wanted to show you today. We are now working on the transnational access, and we are also working on including collection descriptions and including institutional profiles with information about the facilities and the instruments that are available for your access requests. Okay, that's it. Thank you.
All right, thank you, Wouter. We've got a couple of questions for you in the Q&A. First off, we have from Deb Paul, are virtual access funds available for non-EU countries?
Yes, they are. This principle is not restricted to EU countries at the moment.
Okay, great. All right, this one's from David Shorthouse. Have you received any signs that ELViS could be used as a platform to be used outside the EU? And is this how requests for loans for EU specimens will be made from outside the EU?
Well, as I said, ELViS is designed to include collections from Europe, but can be used by anybody in the world. So also people outside Europe can use that to get access to the European collections. There are a few organizations outside Europe that have shown great interest in the development of ELViS. ELViS will be created as open source, and it might be that other continents in the world are also going to use the same solution. Maybe adapted to the local needs, which this can be a little bit different from the situation in Europe.
Great. Is it expected that in the future fundings will be allocated more and more to virtual access instead of physical access to the collections in the synthesis network?
We think that both will be needed in the future, and so both there will be funding for both, and there will not be a bias towards one of them. It's not planned at the moment.
Great. So this is a clarification question. ELViS was built for European collections. Can other collections implement it themselves, taking the development framework? I'm assuming that might be yes, because it might be open source?
Yeah.
Okay, great. More of a clarification question. We have a privacy question. So this person is concerned that requests are public to any user. Privacy might be an issue. Can you speak to that?
In the case of the virtual access calls, we think there's no problem there, because basically it's not the requester who gets money for the request. It's the institute who's doing the digitization who gets the money. So that's why we think we can have this fully open, and it's no problem. And people know from the start that this will be open, and it is the purpose to do so, to have this as a community effort. For transnational access, it's a bit more difficult, because for transnational access, the funding is travel funding that goes to the requester.
Okay. All right, we still have some time. So this is from Robert QB from the chat. So Robert's wondering, if ORCIDs are required, why not use them as the sign-in system? And then Robert provides a link to the integration of ORCID sign-in on ORCID's website.
Yes, this is what we are planning to do next year.
Okay.
But we have not yet implemented this as a first priority, but this is in the planning.
Great. Awesome. I guess we have a couple more moments if you want to get a couple more questions in. Other than that, I think we are through the Q&A.
Okay, great.
We will wait.
Oh,
Is there another question?
there's one more question.
Great, we have time, because I don't want to start the next one until the actual time.
Right. All right, so another question from David Shorthouse. Might there be a pushback from academics who do not yet want to reveal what is the request for fear of being scooped?
Well, the request is only visible after it's submitted for evaluation. So until then, it's draft, and as long as it's draft, it's not visible to anybody else. So in principle, people could keep it as draft for a long time. But of course, it needs to have some time for evaluation by the institutes.
Awesome.
We have not really an indication of that pushback at the moment.
Great. All right, so we still have, I guess, a couple minutes. If you still have more questions, other than that, we will begin our switch to the next presenter.
And just a reminder, if you have questions for any of our previous speakers, you can still put those in the Q&A and they'll be able to see those and type a response.
And I will just wait just a minute or two so that if someone's coming right at 15:30 for this talk, they won't miss the beginning.
I think we need Paul to give us another geology or paleontology joke. He has some good ones.
I don't even have
Paul.
one prepared here. I'll go with the one I did last time. Probably a lot of people didn't hear, and that's a golden one. But a little computer tip for everyone out there. You know how savvy I am on the computer here. But why should you always name your Wi-Fi Yellowstone? Well, because it's a hotspot.
Oh, that's a good
Nothing,
one.
huh? No.
Yeah.
All right.
I mean, the geologists in the crowd like it.
Yeah.
Right?
All right. Maybe.
I did. I loved it. It's good.
I have to say, Paul, I'm missing the dad jokes. I don't know if Jacob is here. Jacob is here. So Jacob Van Veldhuizen, who works with me, there's a couple of us in the paleo section that have a dad joke off, and we just continually trade dad jokes, as we call them. But since we haven't been at work for months and months, I'm missing our dad jokes.
Well, don't expect perfection from geologists. We all have our faults.
Oh, there you go again.
So good. So good.
All right. Well, I think I'll do a nice lengthy introduction for our next speaker, who is Patricia Mergen. And she will be presenting on living collections, so much more than an eye catcher in the landscape. Patricia, thank you. And remember to say next slide, and I'll proceed for you. Thanks.
Okay. Hello, everyone. So I'm presenting on behalf of Elke and Mark, who are our living collection managers in Meize, but who could not be with us today. Next slide. So we will briefly introduce why living collection are eye-catcher, and then touch a bit on the international context, and then look into possibilities that living collections are better taking account in the development in the different initiatives. Next slide. So at Meize Botanical Garden, we have in our 92 hectares, for example, up to 18,000 taxas of living collections. Next slide. So when we talk about definitions of what are living collections, in general, we think of biobanks, zoos, aquaria, culture collection of microorganisms and plants. In the context of botany, we look into seed banks, in vitro culture collections, for example, of algae, fungi or merisderms, and also the indoor and outdoor living collections. Next slide. They have an important role in different areas, like connecting people with plants, celebrating heritage, knowledge transfer, scientific research, and biodiversity conservation. Next slide. As an example of how important they are, for example, for cultural heritage. We can cite that some specimens, as it's the case of preserved specimens, some living specimens have a important historical background, like for example, the mother-in-law tongue plant with the yellow banded, which was only found once in a forest in Africa, and it's believed that the plant that is still multiplied in the garden is at the origin of all the plants that are now existing worldwide from this species. Other specimens are quite unique, where it is believed that there is only one female specimens existing in conservatories, and others have quite nice historical background because they were linked to gifts or donations linked to historical facts from different monarchies or so, and are still present in different botanical gardens. Next slide. So for conservations, for example, the seed banks are quite important for research on crop wild relatives or to conserve plants that are unique and threatened, like the copper flora in Katanga in Congo, or for restoration of degraded sites in different countries. Next slide. Also, the gardens play an important role in the conservation of endangered species or nearly extinct species, or species that are already extinct in the wild, and they play an important role in ex situ conservation. Next slide. And also sometimes, like for the euphorbia, it's quite difficult to keep them as preserved collections in herbaria, but they are very important plants, for example, for health research, for anti-cancer, and it's really needed for studies and research, and also for identifications, to have access to the living plants. Next slide. And then also in terms of food and agriculture, they are very important. And for example, if something happens to our crops, to have different diversity, like for example, in coffee, beans, or bananas, so that if something happened, you can call upon the species that are kept in the botanical gardens to find maybe replacement species for those that are now used economically. At Meze Botanical Garden, since the 90s, we keep databases with our accessions that are more than 36,000 for almost 18,000 taxa. And these databases, next slide, enable that we can do exchanges with different communities, and we get, next slide, we get requests for materials, for seed, DNA samples, plant part for propagation, also fresh samples, sometimes just for information, sometimes coupled with a request for an herbarium specimens, and also about pictures about the plants. Next slide. So in terms of access, for most users that are either from other botanical gardens, internal or external research students, and also artists, the access to this information and material is most of the time open and free. Only when there are commercial users, we make more separate agreements with those partners to see how we can work together to have access to this type of material. Next slide. So in practice, people can either go directly to the website of the garden, or passing via the BGCI, which is the Botanical Gardens Convention International, where they can discover which plant material exists worldwide. And we stress here really the importance to have a good visibility of the collections out there. Next slide. So a bit more words of the Botanical Garden Convention International. So there you have 600 institutional partners from more than 100 countries, actually, with the objective to help in plant conservations, but also to look at policy and advocacy and to support training and capacity building. Next slide. And there you can, via their plant search, find more than 1.4 million collection records with many taxas, and you can locate which species are threatened and how to make material request with their tools. So they use a system called IPEN, the International Plant and Exchange Networks, which tries to respect the regulation of the Convention of Biodiversity and also the famous Nagoya Protocol. And they would like really to help the non-commercial use of plant genetic resources. Next slide. So for that, they issued a code of conduct to respect access and benefit sharing. And they also provide quite some documentations to be able to follow the regulations. Next slide. So this you can look further after. It is an example on how when you collect plant in the wild, for example, from Madagascar, how they make sure that it enters the system and follows at best, the access and benefit sharing regulations. Next slide. 2 slides next, I think. We are a bit behind the
Is
next.
that right?
Yep. And so in the context,
about 1 minute.
Yeah, in the context, for example, of DISCO and CITAF that were presented before, so the living collections like biobanks and seed banks and so on are handled, and they look also at the IPEN system, and there are agreements with BGCI, but when we look at the description of the collections, often the tools presented are more focused on preserved collections, and the needs for having access and seeing the living collections that are part of the network often is not seen in that single access point, and therefore we would like to plea for more collaboration, for example, with BGCI, and that when you enter this DISCO and CITAF structure, you can see the living collections and then get directed eventually to the existing experts. So next slide. So there are already work going on. So for example, the ABCD standard that was also presented in other session earlier today has already concepts for living collections, including the IPEN system. So we don't start from scratch, and it would be good to look in more detail how these concepts can be more integrated, for example, in the Tadwick collection description work that were also presented earlier today. Next slide. So we also plea for more interlinking between preserved and living collections because they can be complementary. And for example, on our portal at the Maison Botanical Garden for Belgian collections, we have now in the same system, you can have access to the herbarium collection and to the living collections. And to conclude, we would just like to have more attention for the living collection, especially the outdoor and indoor higher plant collections, to work more with BGCI. So as I said, contacts and MOUs already exist, but we would like to look also more into using the IPEN system, especially also to help with complying with the Nagoya access benefit sharing and also with the future sequence information regulations, and to be more integrated in the work of the Tadwick collection description group. And for that, it would be useful to have a task force for botanical living collections to work with that group, because they have a lot of work and we need really a core group of experts. So that would be a call if people are interested to collaborate there. And within Coast Mobilize, that is a coast action and European system that is organizing network events. We have foreseen 2 events, one this year online on the IPEN system with my colleagues from Berlin, and then a second one earlier in 2021 that would look with an expert group to see how we can integrate a minimal needed description for the living collection into the ongoing work on collection descriptions. So I think the rest is, the next slides are acknowledgements and context detail. So I can try to answer the questions now, and then you can contact us if you have more questions.
Awesome, thank you, Patricia. We do have a couple questions in the Q&A. First off from Jessica. Jessica's asking, are there tags or labels on plans outside that the public could scan, perhaps with a QR code, to learn more about the specimen?
Well, yes, that happens. I would have to check if we have already implemented it at the garden. I'm not sure, but I know that in some gardens this is actually a possibility. I know we have panels with QR codes where people can get more info, but this is definitely also to have QR codes or other web applications so that people can have more information on the plants on their mobile phone, yes.
Great. Deb Paul asks, do you publish your living collections data to GBIF?
Yes. So for the Mesa Botanical Garden, we have published a data set of the living collections there.
Great. Kyoko asks, any chance to collaborate with partners in Africa? For example, East African herbarium housed at the National Museums of Kenya?
Yes, we already do that because we are behind the flora of Central Africa with several colleagues. So there are probably already contacts, and we are also involved in the GBIF Biodiversity for Development program, where we are mentors and training providers. And we had already colleagues, but that was from the Zoology Museum in Kenya, that came into these programs that I remember. So definitely there are options to collaborate with Africa, as a large part of the collections in our botanical garden are from Africa.
Oh, well, good point of contact, I suppose. All right, this is more of a comment. Dirk Newman points out that IPEN exclusively covers living plants, but not herbarium material or other preserved plant material. Yes. Could you speak to that a little bit?
Yeah, that's true, but there are colleagues in the DISCO and Synthesis program who are looking into this IPEN system to see if it could be adapted also for preserved specimens and for biobanks. There are some issues because the living material is more a dynamic exchange process that needs a bit daily or continuous management. But one of the workshops I explained is exactly to look into this. So the contact is Ole Zeberg and Eva Hefner, who are looking into that, and it's a task within the Synthesis project to look if it's possible to expand IPEN to other material than living plants.
Great. Even fossil specimens?
In DISCO, geodiversity and earth science is also part of it. So this is to look, but as the geology and the earth science specimen actually don't fall directly into Nagoya, but have other regulations we have to see. But there is a specific group in CETAF looking into the earth science collections. So And one thing that is interesting is this ABCD schema I showed before. There is an extension for Earth and Geosciences to ABCD that this group is also looking at. So this is also something where we could work on common ground, because ABCD has this special concept for living collection and also for Earth and Geoscience. So this would help to use maybe the same references and same systems to move ahead with these tasks.
I'll just pipe in here. Holly, I just allowed your talking permissions. I was looking to see if Marika was here, because I think she's helping with some of those initiatives. I don't know if Holly, you want to say anything else?
Can you tell me which initiatives? Sorry.
That's okay. We can move on to the next question. I was looking for Marika, but I didn't see her.
Yeah, Marika is involved in this, and she's looking, for example, in use cases and developments. It's also within the DISCO system, and we work together for the Earth Science on Wouter.
Awesome. Yeah, I've dabbled a little bit with the extension for geosciences for the ABCD schema, and it's pretty comprehensive, and I'd be interested to see more use cases for that system. So yeah, it looks like we are out of questions in the Q&A. We are going to switch over to the next presenter and begin in a couple more minutes, just to give people time to take a breather.
Mark, I'll introduce you in a minute, but you could start your screen sharing now.
Okay, Zoom US will not screen share. Okay, so I just have to, I think it should be good now. PowerPoint, share. Okay, can you see that?
Yep, looks great.
Oh, okay.
I'll give us a minute before I introduce you though.
Sure. It's almost more nerve-wracking and virtual.
I find it a little weird because you can't see any reactions of the audience. You're like, I guess they must have loved all the jokes.
Hope so.
You can't see anyone's faces. I mean, you can see sometimes videos and photos, but it's not the same.
Yeah.
Yes, it's very weird just talking to your computer screen. Unnerving. Sorry.
Paul, I guarantee I wasn't rolling my eyes for your geology jokes too.
You weren't rolling your eyes?
I wasn't, no.
You weren't the one throwing the tomatoes, were you?
No, no.
And I just want to remind everyone, if you still have questions for any of the speakers, you can put those in the Q&A. And the speakers, for you guys, you can open the Q&A and type answers as well to some of those questions if you like.
All right. Well, our next speaker this morning is Mark Smith. His talk is Microscopic to Macroscopic Photography, Bringing the Small to the World. You can go ahead.
Thank you everybody for participating and joining in on this. I know that the conversion to virtual was pretty fast, especially to accommodate Black Lives Matter and STEM and everything. It's been pretty amazing. So I'm going to jump right in because this is probably one of the shorter talks I have to deliver. So I'm Mark Smith. I'm a geologist by training and education. I got my master's from UConn in structural geology, but I was also a big hobbyist and enthusiast in just standard focus stacking back when it was beginning. And my neighbor was a molecular biologist at the US Army who was trying to digitize vectors, problematic vectors like ticks and mosquitoes. And I was able to go to his lab. We were able to work on a system which had then been patented. And then what we tried to do was commercialize it after I graduated. So a lot of this is going to have a geoscientific spin, but also a great spin on the work we do with museums. So at the time, what we were dealing with were 4 primary imaging platforms, SEM and TEM, which are very good at getting very small structures, confocal on a similar light, and then compound microscopy and stereo microscopy. What we were trying to do is really figure out what the limitations of these systems were and improve upon it. And Automontage was already doing that. It's a Leica system where in 2006 through 2010 was really a go-to system for focus stacking. But the optics were not necessarily paired perfectly with the resolution in the camera sensors. And not only that, but the cost was not conducive for standard use. I mean, these systems were over 100K, and that was over a decade ago. So they were not easily accessible by any means. So what we wanted to do was take that hobby background that we had in focus stacking with these home-built platforms. This was actually the first prototype, and try to merge the best of optics and the best of resolution directly with the best cameras on the market. So initially, when we built this, I was doing my undergrad, and a researcher clued in onto my undergraduate research project, which focused on this. And he was a molecular biologist also, and we were looking at Rhizoactinius solani, so barrage line development, how one virus can kill a second virus after it has been mutated. And this is really for defense type systems. It was sponsored by DARPA to protect rice and potato crops. And like I said, this system initially was built in collaboration with the US Department of Health and the Walter Reed Institute. So a huge, huge emphasis on small size vectors. We're talking 1 millimeter and smaller. And then what we tried to do later was really improve upon our magnification capabilities. So the limitations of those conventional imaging systems really revolved around depth of field and a capable platform of capturing color. And what we wanted to do was merge those two, but not through the process of producing a large stereo photograph. We want to take one final image so that the data is manageable, and also have it be very, very high in quality for a wide range of users. And what we really wanted to do was build a portable platform, one that was small, but really took the best of the components, a stable platform that could accommodate a wide range of materials, everything from wet, dry, pin, slide mounted. So we're all familiar with focus stacking. Per the rules, I'm not going to play this video, but really it's a focus stack through this flies, well, it played anyways. But that is, this is the final result. It's a focus stack merged of 200 photographs. And if you notice, one thing we try to do is focus, no glare, any common artifacts in photography we try to eliminate entirely. And when we first built the system and commercialized it in 2013, our limitation was around 100 microns of detail, but we were already shooting slides, and we were already shooting very difficult materials like ambers or other materials that were in alcohol. So the next phase in 2015 was then to create our micro kit, which allows us to capture high magnification data. And this gets us all the way down to 2 to 4 microns in size. So this is a very tiny parasitoid wasp. And then you can see the actual cell structure, very comparable to an SEM, but again, non-destructive, portable system in color, and image capture time in under 3 minutes for something this small, which is very quick when you're 0.5 millimeter or less. The next thing we wanted to do was integrate 3D modeling techniques. So this is actually a 3D model of a very small rodent tooth. You can see it in comparison with the dam or the dime there. And not only that, but also be able to, it's a lot easier to 3D model something larger than it is smaller, but we wanted to build a platform that could do it all. And then we go to a lot of conferences, and because my wife is a biologist and I'm a geologist, we sit and we listen and try to build niche systems for specific problems that researchers and curators are having. So after that, we then switch to a petrographic analyzer, which allows you to cross-polarize slides and materials, but does away with the expensive and limited petrographic microscope. It allows you to use a standard DSLR, and that same platform is also being used by many of our clients to image other slides like Lepidoptera genitalia and so forth. And again, this is just an example of how it's digitizing rocks and minerals in thin section. We then broke into UV light and fluorescent light capabilities, very low cost. We've been able to integrate and 3D print many optical filters that basically adapt directly onto existing flashes. So now you can take the same system and start to utilize ultraviolet and fluorescent light. So these are some examples of a GFP stained mouse embryo and actually looking at the torus structure of blood cells and just a ruby under fluorescent and reflected light microscopy. And again, we're working with a variety of geoscientists. So we also build these automated platforms that focus on digitizing core samples. So sedimentary lacustrine sediments and other heavy duty rock core samples. And then we also broke out and started building a system that could focus on tree core. So this is an entire sample of core and images, not just the rings, but also the cell wall thickness between those rings. We also work with a lot of people to deliver these custom systems as well. So for fast, rapid slide imaging and so forth. And what I really want to talk about are some of the workflows we've been creating with our groups. So early on, some of our clients at the Smithsonian and USDA, Matt Buffington, Lourdes, Jim Woolley, we sat down and started building workflows that were specifically tailored to them. They're looking at parasitoids that are 0.5 millimeter or smaller, but at the, in the same light, a couple of weeks ago, they were digitizing the large murder wasp. So what we're trying to do is be able to build a platform and a workflow that not only helps them with their image capture process, but also the way that they interpret label information and database so that it's standardized across the Hymenoptera platform. And likewise, we've done the same thing. Jennifer Zaspel contacted me a few years ago, and she was putting out this terrestrial parasite tracker grant, which is getting together museum curators for parasite tracking. And again, we focused on a working group. This is us at the Chicago Field Museum. And we were trying to build a workflow. And what's so unique about this workflow is it was initially started to be put together by Alyssa Kaywood, and she did a great job. And then what we did is we sat down and went through and tailored a lot of it. And what it does is everything from capturing the label data, how to interpret the label data and standardize it across multiple platforms such as GBIF and Globi. And then we also have different workflows over here. So for slides, pins, alcohol, wet specimens, but not just tailored for high quality images of type specimens or for evolutionary work, but we also have tables and workflows that are specifically designed for high throughput. So if you're not so focused on high quality, we can bring down and increase the speed quite rapidly. So in the end, what we tried to do was produce a portable system, really one that you could use in the field, but turned out being very useful for people traveling to museums to image type specimens. But also as a geologist, what we tried to do is have it so that now you can take your DEM, georeference, and satellite data and then photograph your field location, the samples from that field location, and then all of the materials, other different types of analytical techniques. So really have one tool that allows you to digitize everything. And I have 1 minute left. So what I wanted to do is just speak to some of the things and ways that we work with other researchers that museums have exhibits to try and push this out. This was an exhibit which is called, it's the microforest. It's mosses and lichens enlarged to 12 feet panels that's going to be installed at the University of Connecticut Natural History Museum and also down in Patagonia where the samples are from. This was another example where we were working with an actual artist, Fiona Partington, where she featured Vladimir Nabokov's work with his blue morph butterflies. And we traveled to different natural history museums to digitize his particular materials. And then these are just exhibits where we were demonstrating the complex symbiotic nature of these people's specific research studies. So here, this is the Hawaiian bobtail squid. This is an ant that produces its own antibacterial own antibacteria medicine to actually treat fungus. And then another panel to describe how plants have sex, and then tapeworms. And then we also did a paleobotany one that what this does is it actually takes fossilized leaves, and they're looking at the stomatal counts to reconstruct what the CO2 conditions were in paleo atmosphere. So with that, I think just a little over 10 minutes, but that is a fast version of my presentation. And again, I want to thank everybody for having me today. I'd be happy to take questions.
All right, thank you, Mark. We do have one question so far in the Q&A. Christina asks, can you explain more about the petrographic analyzer?
Sure. So what we did, it functions the exact same way as a petrographic microscope, except what we've done is we've taken a UV filter and a polarizer, and we mounted that on a platform. This is all on our website too. And if you go to the Meet Our Clients tab, you can actually see other researchers that are using that to case studies. But what it's doing is it actually shines light down onto a white matte base, and then that reprojects light back up. So, and that goes through the bottom polarizer, and then through the slide, and then through a secondary polarizer that's mounted on the lens. So that allows you to measure pleochroism. You can rotate it 360 degrees, but also where a petrographic microscope has a lot of bright hotspots that come up through the crystalline matrix, because we reflect light off of a matte base, we virtually eliminate that. So you're actually getting images from the top and the bottom, you're illuminating it, and it's a totally matte image with no glare. So it's also very good in terms of getting great image clarity.
Awesome, thank you. Allison asks, you mentioned filters for imaging fluorescent specimens and making that part of the system. Could you elaborate on how you find or create those filters?
Yeah, so it's through partnerships like this. So initially, Charlie Maisel, he's with Nightsea. He's a vendor at ESA, most commonly, and he's in Massachusetts. We're in Connecticut. So what he does is he makes the glass for these things, and then buys yellow filters from Tiffin, which are just standard off-the-shelf filters. But the blue glass filters, he cuts them down to size and then sells them to manufacturers like us, that then try to integrate them with whatever our lighting methods are. So we make adapters specifically for the Canon flashes, but we can make them for other flashes as well. And I should emphasize that the workflows that I'm talking about, while they do tailor to the system and platform that we make, they're not limited to it. You can also apply these to the other imaging systems you have, likewise with the fluorescent filters.
Awesome. Thank you very much. Another question from Christina. Could you expand more on 3D modeling small mammal teeth?
Oh, yeah. So that was mammal tooth, or a small rodent tooth, was actually the very first subject that we tested. So basically it's mounted to a pinhead, and we have an automated rotary stage and an aluminum micro gimbal that allows you to rotate your material. We also have it so you can mount it in one axis and rotate it almost so that it's a bit oblique. And that way you don't even need to capture multiple axes. You can just do one round of images in 10-degree increments. And paired with either like a 7.5 or a 10x objective, which we have, that's a perfect scale for small mammal teeth. And then what we use with our product also comes licenses to Agisoft Metashape, now it's called. It used to be Photoscan. And that is the tool that allows you to take the data from our images and then basically produce a 3D model. But because of our significant glare reduction, it's a lot easier to build a model now than it was before, because there isn't as many imaging artifacts that sort of corrupt the file to build the 3D model.
Great. Lauren Smith from the Field Museum asks, well, first they comment that the images are beautiful. Do you have workflows for historic slide-mounted material that may be damaged? For example, slides where cracks or bubbles exist, or the medium has changed color?
Not a specific workflow, but I certainly have certain tips and strategies we have. I would liken that to something like amber. There are ways where you can mess with the refractive index through a certain medium. So I would actually recommend maybe submerging them if you're able to, but if you're not and that's destructive, there are other ways. But I would have to sit down. You are happy to contact me and send me a few samples, and we can work on something where I can just test it and see if there's anything I can put together to help you. I do that for stuff all the time.
Awesome. Thank you very much. Have you studied the property of pleochroism by petrographic microscope?
I wouldn't say I studied a particular field. So for my undergraduate research, what I had to do was look at an earthquake zone through Taiwan, and we looked at examples of cataclasis. So rock that's very broken from earthquakes, and we had to reconstruct where the stresses were in the earth. And for that, pleochroism is important, but it's more important for mineral identification or fluid inclusion, like the petroleum industry. And what happens is when you rotate a slide, the minerals will go extinct at certain angles, and that helps identify the exact mineral that you're focused on or looking at. So that is usually how pleochroism is studied. There are 2 handbooks that I use as resources, and It's been a while since undergrad, but I do have those books, and I can't remember what they are offhand, but what I can do is I can go in the other room, look at them, and I can write them in the comment section.
All right, so we have a few clarification questions. So Martin asks, could you repeat the name of the museum that will have the microscopic forests exhibit?
Oh yeah, so that's the University of Connecticut Natural History Museum. My wife's a adjunct faculty there, and I'm a grad. It's right down the road, and it's also going to be it was installed at the embassy, the Chilean embassy in Washington D.C., and it's also going to be installed later on at a new museum that's being built it's either new or existing, kind of going through secondary information here, but this is in Patagonia. So it's going to be installed at 3 different places, but it's going to be built as a traveling exhibit, so it can go other places as well.
Okay, cool. There are a couple more clarification questions, but they're more like looking at links, so I'll leave that to you later. We have just one more question for your presentation. Magnesia asks, has your petrographic setup been used for visualizing fossil insects?
So it can be used. You can place an amber on top of it. What you really want to try to do is look at more reflective light for that type of material. I guess the short answer real quick is no. We usually use other methods to look and examine that type of material. I'll leave it at that. All
right, great, thank you. Perfect, so if you would direct your attention to those links in the Q&A, you can see them, correct? Cool.
Yep.
I'll leave those up to you, and I'll get the next presentation ready, and we'll start back up at 12:10.
Thank you.
Thank you, Mark.
Will we just be running the video?
Yes.
Yes, oh, sorry.
Oh, it's all right.
I have the video. We'll wait a couple more minutes, and then I'll get it started up. And then Constance, I assume you're here for a Q&A afterwards?
And so is David Eagledon.
Okay.
And I'm not sure if Jane made it or not yet.
Okay.
Haven't seen Jane, but I've been checking.
I think Jane might be Jane S5.
Oh, perfect. I will promote.
Thanks.
Well, I guess I'll just mute during the video.
Hello, yes, I'm here. Good to see everybody. This is Jane.
Well, welcome. We have just about a minute to go.
If you're just joining us, just a reminder to direct your questions to the Q&A, and then we'll address them at the end of the presentation. Get started in just a couple moments. Okay, I'm gonna start sharing my screen, make sure everybody can hear everything. So get started. Do the full screen.
Hello, I'm Constance Rinaldo, the librarian of the Ernst Mayr Library of the Museum of Comparative Zoology at Harvard University.
Does the sound work for everybody?
Sounds good to me.
Awesome. I will continue.
I'm also the chair of the Biodiversity Heritage Library Members Council. I'm joined by my colleagues Jane Smith and David Eagledon, who will introduce themselves.
Hello, I'm Jane Smith, Head of Library and Archives at the Natural History Museum in London.
Hello, I'm David Eagledon, and I'm Head of Data and Digital in the Library, Arts and Archives, the Royal Botanic Gardens Kew in the UK.
Our talk today is about how libraries and archives are working together to support the natural history sciences. Natural History Museum's key assets beyond their staff are their collections, the specimens, libraries, and archives. Connecting these assets will contribute to the understanding of biodiversity in the uncertain world we are currently in. Natural history collections, including library and archives collections, can date from the 15th century to the present day. The library and archives collections have been developed to support and complement the specimen collections and the work of the curators and the researchers. And for them, even the ancient data is still useful. Biodiversity literature and archives are scientific data. The Biodiversity Heritage Library contains data that's critical to studying life on Earth and can contribute to the discovery of how the loss and misuse of biodiversity can generate undesirable economic, sociological, and health consequences. These data found in libraries and museums all over the world, but they're disconnected. So our job was to pull them together and make them available to all. Some of the rich data sources, such as species descriptions, distribution records, and other things listed here, are critical to looking at climate changes over time and historical scientific observations. And being able to connect all of these things together is one of our goals. In order to do this, we created the Biodiversity Heritage Library. As noted at Biodiversity Next in 2019, we're all looking for better science, better collaboration, and better data. The BHL is a digital library and a data resource. The key driver is to meet the needs of scientists who rely on historical and current content to complete their research. It's multidisciplinary, it's virtual, it's open access, and it's a collaboration with content from 1450 to 2020. It's international in scope, and the members' collections complement each other. We also have a number of services that support data extraction and sharing. BHL's natural ecosystem is that it's virtual, it's global, the partners collaborate and are interconnected. BHL is resilient, and the partners digitize for open access with the overall theme of open science. We have a range of free services, API and data exports, taxonomic name finding, downloading of PDFs, OCR, and JPEGs, article indexing, and assigning DOIs. You can look at collections in BHL in a variety of ways. We can curate content by theme and also by contributor. In other words, each contributor has a collection that highlights its own contributions to BHL. BHL has quite a lot of legacy literature, but also it has started to add more current literature. We're chipping away at it. The content we need now is an increased amount of current materials, as well as adding original manuscript and field notes. Through citizen science opportunities, we can improve the accessibility of data within BHL. And you can see here a couple of examples using transcription or tagging Flickr images. If you'll note that the red line is highlighting the information about taxonomic data that was added by volunteer transcribers, volunteer taggers, to the Flickr image information. And here you can see a broader view of some of the unique content that we have in the Biodiversity Heritage Library. This is a page from Field Notes, and the page has been transcribed, as you can see on the right. And by having this page transcribed, we can now extract the taxonomic information from this page, as well as search this page within BHL. And this is just an example of searching the page within BHL. When you search for a taxonomic name, for instance, in BHL, you can bring up a bibliography. And this particular bibliography shows you that pages from Field Notes and Diaries, the long list of journals of William Brewster at the bottom, can now be incorporated into these searches. Volunteer transcribers make this possible. So BHL has moved from a digital library to a data resource. We're connecting information. We're opening up all the various possibilities of information scattered around and being able to put them together. By digitizing, depositing, linking, transcribing, we can extract more data and then repurpose this data. You can unite resources within an institution. You can see here a variety of slides of pieces of information, observations and notes, later digitized and transcribed for BHL, original specimen record, and the publication of the species description, and an image that was digitized for BHL. And these can all be collated and put into a local record. And here's the local record itself. So we have bits and pieces that are all added into this local record, and this local information, the local data, can then be shared globally to aggregators so that it's available to anybody. Another advantage is that you can unite collections from multiple institutions. So different institutions deposited correspondence from into the BHL, and the letters to and from a particular individual can be put together and shown side by side, even though they're in different repositories. So why should we keep contributing? Why is BHL important to individual institutions? Some of the reasons provided by our partners, really overall, it's about supporting science. It's about opening access. We can deliver content, we can share content, and we can provide curated content. And this content can be linked. And when we have all this content available, we can expand our local and global impact as an institution. We also provide more network opportunities. Another important point is that some institutions use the Biodiversity Heritage Library as a repository for all of their information. So BHL is part of a community in which all players have benefited by making original materials accessible and have contributed data as an integrated part of all the global biodiversity initiatives and organizations. And of course, in this current climate, social distancing is what BHL does. BHL collaborates virtually and it opens up science. And this is the natural ecosystem for the institutions that have been working with BHL. The cumulative effect of our efforts working remotely and supporting large-scale science, we hope will help solve planetary problems that are in evidence now, particularly COVID-19. Thank you so much for having us here at this conference to be able to talk about what is important to us in terms of making the libraries and archives materials more broadly available. BHL is working on some future directions to improve scientific name finding, working through the global names architecture, also improving data links with object identifiers so that we can drill down to pages, to scientific names, and maybe even more. If you have any questions, we'd be happy to field any questions through our email, or we're all on we're here even if we've recorded this session, so you can ask us directly. Thank you very much.
That was great, Connie, and I'm sure most of us have appreciated BHL throughout our careers. We do have a couple questions. The first is to Connie and the co-speakers. Will we be able to link specimen data to BHL resources and vice versa? Can the public, all of us, help with such an effort?
I'll start by answering that. We're working on ways to identify a much more granular set of information. And certainly you can, if you can find it right now, possible, but like it's, you know, it's full text searching. If you can find it, you can certainly link. And if we don't currently do it the other way around, but that is certainly a possibility if we are able to use some kind of digital object identifier to tag particular items. And currently we don't have the option of, it has to be someone, a member from BHL who does that kind of tagging. At some point we are hoping to have more annotation possibilities, which would then facilitate the process. Jane, do you want
I did. I'm sure there's a way of putting your hand up virtually here, but I can't figure it out at the moment. So Connie's right in terms of that volunteer help in tagging, but I think probably what's developing is through in individual institutions that volunteer help is coming through as well.
If you've got any other questions, you could type them into the Q&A.
I just want to note there's a lot of love for BHL going on in the chat right now.
I've been seeing that, and that's really nice to see. It's a difficult time right now, and it's good to know that I mean, that was really what we were thinking, is BHL can help a lot in terms of some of the work that people have to do remotely. And I know that's true in the MCZ because I've heard that from people. So, and if you have ideas or requests, feel free to get in touch with us. There's a feedback link in BHL, and we do read all that feedback. So anything you want to suggest or have to say, ideas, please feel free to use it, and it will somehow come through our pipeline.
Great.
Yes, I just wanted to add actually, thank you again for all the comments. It's lovely to see them, and do send them in to us via the BHL site. We collect them all and read them all, and you know, they're really, really useful testimonials for us to underline, you know, how useful and relevant BHL remains. So please do.
And we've got a couple more questions in here. This one's from Brad Scott. The last one was from Deb Paul. I forgot to mention that. Brad asks, is it possible to import manuscript transcripts captured in DocBook or TEI into BHL?
That's a little too specific for right now. However, we are working on we are working on getting born digital materials into BHL. Again, it's not just a general, a member has to do the importing basically, or some kind of partner who has access to the administrative functions of BHL. And we're just now in the phase where we're able to add, as I said, born digital materials. We haven't been able to do that prior to this, and part of that has to do with just our material is all housed at the Internet Archive, and the protocol for how that works has been a bit of a problem with born digital materials, but we're working it out now. So that's a question that I can't answer right now, but it's definitely a good one for the future.
All right, and another question that every speaker hopes will be asked during their presentation from Lauren Gardiner: how can organizations get more involved in BHL?
Well, we've typically run as a member organization, And right at the moment, we're closed to members because we've had, we didn't have a program manager for over a year. And we just ramped up a new program manager. And what that means is there's a lot of training and hand-holding involved in getting people onboarded at BHL. It is a membership institution. We do have a membership fee, and we're actually sort of reworking how that works. So just get in touch with us. You can do it again through the feedback, or you can directly contact Jane, David, or myself, and we can let you know what's happening.
All right, and from Siobhan Leachman, will OCR correction be offered on BHL at some point?
That's definitely on our menu of desires, and it is. I believe it's in our current 5-year plan that we just finished developing. Yes, Jane says yes. It's not up for this year, but it is coming. So within that 5-year period, we should have more work done on OCR. And again, that has to do with, I mentioned the annotation. We do currently add transcriptions, as you saw in the talk, but we add them separately. They're not something that's done in BHL. So we don't currently have a mechanism for people to correct text that's within BHL, but we're hoping to have a mechanism that has some kind of ability to allow you to do some correction, do some transcription, and then that would be downloaded at a different point, but would still be you could do it from wherever you're working in BHL. We aren't there yet, though.
Connie, is it worth saying, you've mentioned our new strategic phase, which is this year until 2025, obviously a little bit disrupted by what's happening with the COVID situation, but we'll be working over coming months on our implementation plan. So it'll be a bit more obvious what our roadmap is for some of these things. Some of them we can only do one at a time because we're reliant on us all giving volunteer help to this, but there is a roadmap.
We do have a very limited technical, you can't even call it a group. So we have one person who does a lot of technical things on BHL, and then someone else who has another full-time job. So we only have one technical person who works more or less full-time on BHL. So we do have to plan very carefully.
Okay, there's just one more question, it looks like, from a digitization intern, Paula Bartow. She wanted to know, how is BHL different from other museum database servers like Arctos and GBIF? Can you speak to the nuances of purpose or limitations and advantages of the different databases?
BHL is a different database. It is built with highly connected to the Internet Archive. It was built around books and journals. Actually, it was built around books, which is what the Internet Archive was built around. And we've kind of managed to kind of squeeze in journals and figure out ways to identify articles, but that's been a struggle. That doesn't mean that the data can't go into those places. We are working on depositing data in GBIF right now, which will probably revolve around images, because we happen to have a bunch of tagged images that are relatively easy to extract. Why did I say that? Not relatively easy to extract, but they're extractable. Sorry, you said something else. Arctos.
It might be worth saying that they're complementary databases, right, and that what BHL brings to I think we're describing it as an ecosystem. We provide the literature and the original materials, so it's probably best to think of us in terms of a virtual library. And just as virtual libraries are developing all sorts of content, and we're very much involved in data extraction that supports a whole range of research and uses. So So there is a little bit of overlap in the middle, but it's very much about being complementary, and that we're providing access to information sources that directly support some of the initiatives that are happening through those other groups.
And just, I did show the Museum of Comparative Zoology database, which has quite a lot of BHL links in it. Most of that, some of that is done through using automation, but a lot of that is done by hand. So we don't yet have a really good way to do that automatically without a lot of human input. So.
I think Jane's point is very valuable, and that they're complementary and that it sits alongside really. But also, of course, there is the added advantage at the moment that we don't have access to our physical collections and libraries and archives. It is a wonderful portal to connect people with them. So there's a lot of advantages there.
Great. Well, thank you all. That was fantastic. And thank you to all of the speakers in this session. I appreciate that you showed up, and especially would like to thank the organizers of this conference who pulled together such a seamless production at very, very short notice, and which has unprecedented success in number of participants, so especially admirable.